Transcription and translation. Transcription. Transcription is a process in which ribonucleic acid is synthesized from DNA. The genetic information stored in DNA is expressed through RNA. For this purpose, one of the two strands of DNA serves as a template and produces working copies of RNA molecules. RNA polymerase. RNA polymerase being the key enzyme in transcription in eukaryotes, several different enzymes are required to synthesize the different types of RNA. They are called as RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. The product formed in transcription is referred to as primary transcript, which are inactive in nature. Alterations like splicing, terminal additions, and base modifications, etc., are called post-transcriptional modifications. They'll produce functionally active RNA molecules. Stages of transcription. The process of transcription can be divided into 4 stages. Formation of transcription complex of DNA and RNA polymerase. The second one, initiation. Third one, elongation. And fourth one, termination. This is a schematic representation of transcription complex, which include the RNA polymerase enzyme, the activators, enhancers, activators, coactivators, and the basal factors. Initiation. RNA polymerase bind to a DNA at promoter region. This is prerequisite for the transcription to start. There are 2 base sequences on the coding DNA strand, which the sigma factor of RNA polymerase 1, 2, or 3 can recognize for initiation of transcription. Specialized sequences of DNA known as Tata box or Hogness box and cat box are being identified by these respective enzymes. This is the Tata box that lies in the core promoter region. Altogether, it forms the initiation complex. The Tata box or Hogness box lies almost 25 base pair apart from the start of transcription, and cat box lies almost 70 base pairs apart from the start point of transcription in the coding region. For the initiation of transcription, there are 3 stages. First one, chromatin containing the promoter sequence made accessible to the transcription machinery. Second one, binding of transcription factors like transcription factor 2D, transcription factor 2A, transcription factor 2B, transcription factor 2F, transcription factors 2E, transcription factors 2H to DNA sequences in the promoter region. It is stipulated, or it is postulated, that the transcription factors binds to each other and in turn to the enzyme RNA polymerase. You may refer the diagram represented here. These are all connected to the RNA polymerase enzyme. Basal factors, activators, the enhancer region, the DNA, the coactivators, all are connected to the RNA polymerase. Stimulation of transcription by enhancers. Enhancer can increase gene expression. This is made possible by binding of enhancers to transcription factors to form the activators. These are the activators, where the enhancers attached to the activators and give signals to the basal factors via the coactivators and to the RNA polymerase 2 enzyme. The primary mRNA transcript produced by RNA polymerase 2 in eukaryotes is often referred to as heterogeneous nuclear RNA, or hnRNA. This is then processed to produce mRNA needed for protein synthesis. This is the nascent RNA or the heteronuclear RNA or hnRNA. After initiation and chain elongation, there is termination of transcription. The termination is with a rho-dependent factor or rho-independent factor, or also known as intrinsic factor. Rho-dependent termination. The specific signals are recognized by a termination protein, the rho factor. Attachment of rho factor is ATP-dependent process. When it attaches to the DNA, the RNA polymerase cannot move further, so the enzyme dissociates from DNA and consequently newly formed mRNA is released. Once the mRNA is released, it will be undergoing post-transcriptional modifications to become the mature RNA. The second type of termination is rho-independent or intrinsic termination. A guanine cytosine-rich palindrome or GC-rich palindrome sequence precedes the sequence of 6 to 7 uracil residues in the RNA chain. As a result, a stem and a loop structure is formed, which is crucial for termination. This is the GC-rich area accompanied by uracil moieties. And it will form stem and loop structure, and then it will be cut off from the transcription complex. And after it is released, the immature mRNA will be, or immature RNA will be converted into a mature RNA with post-transcriptional modifications. This is the GC-rich area and the hairpin loop and the uracil-rich sequence, also known as the poly-U tail or poly-U site. The next is translation. The genetic information stored in DNA is passed on to RNA through transcription, and ultimately it is expressed in the language of proteins. The biosynthesis of a protein or a polypeptide in a living cell is referred to as translation. The sequence of amino acids in the protein synthesized is determined by the nucleotide-based sequence of mRNA. There are wide variations in the cells with respect to the quality and quantity of proteins synthesized. This largely depends on the need and ability of the cells. Outline of translation processes. The first one is requirement of the components. Second one, activation of amino acids. Third one, protein synthesis proper. And it includes 3 subcategories: the initiation of translation, the elongation of translation, and the termination of translation. And again, this is subclassified. Initiation of translation is subclassified as ribosomal dissociation, formation of 43S pre-initiation complex, formation of 48S initiation complex, formation of 80S initiation complex. Elongation of translation is again subclassified as binding of amino acid tRNA to A site, peptide bond formation, and third one, translocation. Apart from these 3 processes, some additional processes are there like chaperones and protein folding, and finally the post-translation modifications. So ultimately leading to the synthesis of a mature mRNA or tRNA or ribosomal RNA. That is the functional RNAs. The protein biosynthesis. Protein synthesis, which involves the translation of nucleotide-based sequence of mRNA into language of amino acid sequence, may be divided into following stages: requirement of the components, activation of amino acids, protein synthesis proper, chaperones and protein folding, and the post-translation modifications. These are the basic steps through which the protein synthesis occurs. First one, requirement of the components. Some of the essential components required for the translation to occur is the first one, amino acid, second, ribosomes, third one, messenger RNA, fourth one, transfer RNA, energy sources in the form of ATP and GTP, and the protein factors. First one, amino acids. Proteins are polymers of amino acids. Of the 20 amino acids found in protein structure, half of them, about 10, can be synthesized by man. About 10 essential amino acids had to be provided through the diet. A regular dietary supply of essential amino acids in sufficient quantities should be maintained, as it is a prerequisite for protein synthesis. Second one, ribosomes. The functionally active ribosomes are the centers or factories for protein synthesis. Ribosomes are huge, complex structures. 70 S for prokaryotes and 80 S for eukaryotes of proteins and ribosomal RNAs. Each ribosome consists of two subunits, one big and one small. This is the large or big subunit, and this is the small subunit. The functional ribosome has two sites, A and P site. A site, this A site and this is P site. Each site covers both subunits. A site is for binding of amino acid tRNA, and P site is for binding peptidyl tRNA during the course of translation. In case of eukaryotes, there is yet another site called exit site or E site. Thus, eukaryotes contain three sites, A, P, and E on the ribosomes. The ribosomes are located in cytosomal fraction of the cell. They are found in association with the rough endoplasmic reticulum to form clusters, rough endoplasmic reticulum of ribosomes, where the protein synthesis occurs. This is the ribosome, larger subunit and smaller subunit. The messenger RNA or mRNA. The specific information required for the synthesis of a given protein is present in the mRNA. The DNA has passed on the genetic information in the form of codons to mRNA to translate into a protein sequence. Fourth one, tRNA or transfer RNA. They carry the amino acids and hand them over to the growing peptide chain. The amino acid is covalently bound to tRNA at 3' end. Each tRNA has a 3 nucleotide-based sequence, the anticodon, which is responsible to recognize the codon complementary basis of mRNA for protein synthesis. Fifth one, energy sources. Both ATP and GTP are required for the supply of energy in protein synthesis. Some of the reactions involved the breakdown of ATP or GTP respectively to AMP and GMP with the liberation of pyrophosphate. Each one of these reactions consumes 2 high-energy phosphates, equivalent to about 2 ATP. Sixth one, protein factors. The process of translation involves a number of protein factors. These are needed for initiation, elongation, and termination of protein synthesis. The protein factors are more complex in eukaryotes than compared to the prokaryotes. Activation of amino acid. The amino acids are activated and attached to tRNA in a 2-step reaction. A group of enzymes, namely amino acid tRNA synthetases, are required for this process. These enzymes are highly specific for the amino acid and the corresponding tRNA. The amino acid is first attached to the enzyme utilizing ATP to form enzyme AMP amino acid complex. The amino acid is then transferred to 3' end of the tRNA to form aminoacyl tRNA. This is the amino acid tRNA. Codon anticodon recognition. The codon of the mRNA is recognized by the anticodon of tRNA. This is the codon that is present in the mRNA, and this is the anticodon that is present in the tRNA. They pair with each other in antiparallel direction, that is 5', 3' of mRNA and 3', 5' of tRNA. This is mRNA 5', 3' direction, and this is tRNA from 3', 5' direction. Third step, protein synthesis proper. The protein or polypeptide synthesis occurs on the ribosomes. The mRNA is read in the 5', 3' direction, and the polypeptide synthesis proceeds from N-terminal end to C-terminal end. In eukaryotic organisms, transcription occurs in the nucleus, whereas translation takes place in the cytosol. Translation process is divided into 3 stages: initiation, elongation, and termination. Initiation of translation. The initiation of translation in eukaryotes is complex, involving at least 10 eukaryotic initiation factors. Some of the eukaryotic initiation factors contain multiple subunits, almost sometimes 3 to 8 subunits may be present. The process of translation initiation can be divided into 4 steps: ribosomal dissociation, formation of 48S pre-initiation complex, formation of 48S initiation complex, and finally the formation of ATS initiation complex. All this is represented here. The formation of eukaryotic initiation complexes: 4F, 4A, 4B, 4E, 2C, the ribosomal dissociation complex. The ATS ribosome dissociates to form 40S and 60S subunits. 2 initiating factors, namely eukaryotic initiation factor 3 and eukaryotic initiation factor 1A, bind to the newly formed 40S subunit and thereby block its reassociation with 60S subunit. Formation of 43S pre-initiation complex. A ternary complex containing met tRNA initiation and eukaryotic initiation factor 2 bound to GTP attaches to the 40S ribosomal subunit to form 43 pre-initiation complex. The presence of eukaryotic initiation factor 3 and eukaryotic initiation factor 1A stabilizes this complex. Formation of 48S initiation complex. The binding of mRNA to 43S pre-initiation complex results in the formation of 48S initiation complex through the intermediate 43S initiation complex. This, however, involves certain interactions between some of the eukaryotic initiation factors and activation of mRNA. Eukaryotic initiation factors for F complex is formed by the association of eukaryotic initiation factor for G, eukaryotic initiation factor for F with eukaryotic initiation factor for E. The so formed eukaryotic initiation factor for F binds to the cap of the mRNA. Then eukaryotic initiation factor 4A and eukaryotic initiation factor 4B bind to mRNA and reduce its complex structure. This mRNA is then transferred to 43S complex. For the appropriate association of 43S pre-initiation complex with mRNA, energy has to be supplied by ATP. Recognition of initiation codon. The ribosomal initiation complex scans the mRNA for the identification of appropriate initiation codon. 5' AUG is the initiation codon, and its recognition is facilitated by a specific sequence of nucleotides surrounding it. Formation of 48 is an initiation complex. 48S initiation complex bind to 60S ribosomal subunit to form 80S initiation complex. The binding involves the hydrolysis of GTP bound to eukaryotic initiation factor 2. This step is facilitated by the involvement of eukaryotic initiation factor 2, or eukaryotic initiation factor 5. As the ATS complex is formed, the initiation factors bound to 48S initiation complex are released and recycled. The activation of eukaryotic initiation factor 2 requires eukaryotic initiation factor 2B, also called as guanine nucleotide exchange factor, and the GTP. The activated eukaryotic initiation factor 2 that is bound to GTP requires eukaryotic initiation factor 2C to form the ternary complex. The whole process of initiation and dissociation of ribosomes is represented in this diagram. ATS and 40S ribosome dissociation and consequent attachment of the factors, initiation factors. Elongation of translation. Ribosomes elongate the polypeptide chain by sequential addition of amino acids. The amino acid sequence is determined by the order of the codons in the specific mRNA. Elongation, a select process involving certain elongation factors, may be divided into 3 steps, like binding of amino acid tRNA to A site, peptide bond formation, and the third one, translocation. First, on binding of amino acid tRNA to A site. The ATS initiation complex contains met tRNA initiation in the P site, and the A site is free. Another amino acid tRNA is placed in the A site. This requires proper codon recognition on the mRNA and involvement of elongation factor 1A and supply of energy by GTP. As the amino acid tRNA is placed in the A site, elongation factor 1A and GTP are recycled to bring another amino acid tRNA. Peptide bond formation. The enzyme peptide transferase enzyme catalyzes the formation of peptide bond. Here, the peptide bond is formed between the amino acids. The activity of this enzyme lies on 28S RNA of 60S ribosomal unit. It is therefore the RNA and not protein referred to as ribosome that catalyzes the peptide bond formation. As the amino acid in the amino acid tRNA is already activated, no additional energy is required for peptide bond formation. The net result of peptide bond formation is the attachment of the growing peptide chain to the tRNA in the A site. Translocation. As the peptide bond formation occurs, the ribosome moves to the next codon of the mRNA towards the 3' end. This process is called translocation. Basically involves the movement of growing peptide chain from A site to P site. Translocation requires elongation factor 2 and GTP. GTP gets hydrolyzed and supplies energy to move mRNA. Elongation factor 2 and GTP complex recycles for translocation. Another site, namely exit site, has been identified in eukaryotes. The deacylated tRNA moves into the E site from where it leaves the ribosome. Incorporation of amino acids. It is estimated that about 6 amino acids per second are incorporated during the course of elongation of translation in eukaryotes. In case of prokaryotes, as many as 20 amino acids can be incorporated per second. Thus, the process of protein or polypeptide synthesis in translation occurs with great speed and accuracy. Termination of translation. After several cycles of elongation, incorporating amino acids and the formation of specific protein or polypeptide molecule, one of the stop or termination signal, namely UAA or UAG and UCA, terminates the growing polypeptide. The termination codon, which act as a stop signals, do not have specific tRNAs to bind. As the termination codon occupies the ribosomal A site, the release factor, namely eukaryotic releasing factor recognizes the stop signal. Eukaryote releasing factor GTP complex in association with the enzyme polypeptide peptidyl transferase cleaves the peptide bond between the polypeptide and the tRNA occupying P site. In this reaction, a water molecule instead of amino acid is added. This hydrolysis releases the protein and tRNA from the P site. The 80S ribosome dissociates to form 40S and 60S subunit, which are recycled, and the mRNA is also released. The whole steps are represented in this schematic representation: chain elongation and termination.